Welcome to our 8th video on mitosis. In our last video, we talked about the stage called Anaphase. We went over some of the interplay and regulation processes of the Anaphase promoting complex, Securin, and separase, and how chromosomes they get separated. That we discussed in detail. If you missed that particular video, it's a good idea that you watch that video before this one. Today, we are going to talk about the next stage, that is the telophase. Let's get started. The final act of mitosis in cell division is played out in telophase. Here, the events of prophase and prometaphase are reversed, leading to the reformation of nucleolus and nuclear membrane. It's a brief moment in the grand scheme of things, taking up just 2% of the cell cycle's total duration. As the chromosomes reach near the poles of the cell, the nuclear envelope is reconstructed around each group of chromatids. Simultaneously, the nuclei reappears and the chromosomes start to loosen from their condensed stage, reverting back to the interphase chromatin form. In telophase, the mitotic spindle breaks down and its microtubules are also decomposed. Cytokinesis usually starts before the late stage of telophase and upon completion splits the two nuclei into separate daughter cells. Now let's try to understand the molecular mechanism of telophase. Here we need to understand what are M-Cdks, mitotic cyclin-dependent kinases. These are also known as M-Cdks. They are the group of protein kinases known for their critical role in controlling cell cycle. In addition to that, they play a part in regulating transcription, cell differentiation, and also mRNA processing. The activation of mitotic cyclin-dependent kinases occurs through phosphorylation. This process triggers key events in early mitosis, such as chromosome condensation, spindle assembly, and the breakdown of the nuclear envelope. On the other hand, the dephosphorylation of these proteins in telophase leads to the disassembly of the spindle, decondensation of the chromosomes, and the formation of daughter nuclei. The deactivation of Cdk is caused by the degradation of cyclin. The Anaphase promoting complex, which is APC, and again it's also known as Cyclosome, targets cyclins for degradation through proteolysis. The separation of chromosomes from the metaphase plate during Anaphase can activate spatial signals that initiate telophase. CDC48, a protein with the ability to alter target protein configuration through its ATPase activity. It is a crucial regulator and driver of telophase stage. It plays a critical role in the disassembly of the spindle, assembly of the nuclear envelope, and the decondensation of the chromosomes. Alright, these were some of the key points of telophase. We discussed in detail about the telophase features, what are the structures that reappear in the telophase. In addition to that, we discussed about mitotic spindle-dependent kinases. Next, we will discuss the stage called cytokinesis. If you liked and enjoyed these videos, please do not forget to subscribe to the channel. Thank you. Let's move on to the next video.